Oh, all right, everybody. Hope you had time for a nice lunch. Always feel like a little bit rushed, but is what it is. Hopefully today won't take too long to cover what we need to cover. Here we go. So before we go on to that, does anybody have any questions or issues or whatever about the Mendelian inheritance lab, which is due tonight? Now's a good time to, sorry, not speaking intentionally slowly, just doing stuff at the same time. Good time to ask, go through anything that you need to go through. Give a few moments for people to roll in and gather their thoughts. All right, well, if any do pop up and crystallize in your brain while we're talking about the stuff, just feel free to interrupt and we'll cover those first because it'll be a little bit more straightforward. Okay, so what we're going to be doing for the next 3 labs, let me just double check the schedule again. We do where that's gone. Got too many Word documents over. Yeah, so essentially for this week and Tuesday of next week, we'll be talking about or looking at a linkage experiment, right? And so one of the reasons why we do this is it's actually well, in real life, you'd be doing a genetic cross between 2 different genotypes of flies to be able to calculate linkage between them. And so right now, because we haven't started linkage in class, we're going to do that on Thursday. This might be a little bit like, what the hell is he talking about? So I'm going to give you a little bit of background, kind of give you an overview of what you're going to be doing and point you to where the data are. And we'll talk about deadlines and assignments and stuff like that. But also because we will be spending a bunch of time talking about linkage, this is a really, really good fit in terms of this is the theory and bless you, here's the practice, right? And so that's one of the big pluses of doing this particular lab. Now, obviously, you're not going to get the benefit of doing the actual mechanics of working with flies, which for some people is not that much of a downside, other people is. But you will get all of the analytical skills, which are really the point that we want to teach you. Plus, you'll also get practice of writing a little mini lab report too, which is a it's a very important skill to develop, which, you probably have some experience already, but you will have more so after this. Okay, so let's share screen so that, oh, look at that, there's schedule. I was just checking to see when the next exam was. Here we are. So basically we are going to be looking at flies and we're going to be looking at 2 different traits. One is eye color, which you can see here. So normally wild type flies have red eyes And actually, if you catch any in your kitchen or whatever, like hovering around your trash can, they will have red eyes on them, guaranteed. Almost, yeah, pretty damn sure they will. And Thomas Hunt Morgan, who's really the true father of genetics, he isolated a Drosophila mutant that had white eyes. Now, through a bunch of cool crosses and experimentation, he discovered that that was a sex-linked trait, and as a result, that gene is on the X chromosome. So that was the very, very first X-linked or sex-linked trait ever discovered, which was pretty cool. Now, we also have another trait which is encoded by a single gene on the X chromosome, and that is a pigmentation trait. We're going to be looking at this in a lot more depth. This is called the yellow or yellow gene, or the body color trait. And we'll be going into the molecular biology and the bioinformatics of this in quite considerable detail after we finish this linkage experiment. So this is also, in a sense, a kind of an introduction for that too. Now, the yellow gene encodes a enzyme which, is it serotonin? I'd have to double check. Honestly, like stuff falls out my head like it's got a giant leaky hole in the bottom. But it's involved in the generation of pigment. And so in a homozygous recessive mutant, or if it's a male in a hemizygous recessive mutant, and I keep saying that, actually I probably better write that down because, accent and stuff may not get that. It's just I guess I'm just showing off really. So homozygous recessive, is easy. So that would be genotype AA, let's say. Hemizygous equals A, because you only have one copy of that allele because that gene happens to be on the X chromosome. And so both of the traits that we're looking at are encoded by genes on the X chromosome. So anyway, if you're homozygous or hemizygous recessive for the yellow gene, you will have a non-functioning yellow gene, and that will result in you having a yellow body. Typically, genes are named after the phenotypes that result from them being mutated and not working. That's kind of the common way of naming genes. They just take it to ridiculous extremes for Drosophila. So you can look at these flies. I work on worms. You look at these flies and you can tell based on their color what their phenotype is for the yellow body trait. These ones at the bottom are yellow, they're less pigmented. You can also see their butts aren't quite as dark. That's another way of telling them apart, whereas the wild type ones are. So linkage is when two genes are close enough on the same chromosome that recombination doesn't always occur between them. So we talk about recombination in meiosis, always happens. At least one arm, if not both arms, have a chiasmata occurring, right, for the crossover, for the recombination. And that where that crossover occurs is random. And this is an important point to get across. Right. Now, if you have two genes close enough together on the same chromosome, there is a probability, because again, it's always a mathematical probability because it's not a certainty, because it's random, right? That recombination will not always occur between them. In that situation, those genes are linked. Ergo, and so the process that we're looking at is called linkage. And so really this is a way of figuring out how far apart two genes are physically, more or less, kind of maps pretty well depending on whereabouts we're looking at, on the same chromosome, and trying to figure out how far apart exactly they are. So this is exactly the kind of work that Thomas Hunt Morgan did to map out the X chromosome in Drosophila. There's a whole bunch of different genes on the X chromosome, and he isolated mutants in as many of them as he could find so that he could figure out their relative distance from each other, doing the kind of experiment that we're going to do. Anybody have questions so far? Okay, so this is going to involve a little bit of funky writing, I guess, in terms of writing stuff out. And everybody does this a little bit differently, but I have my own personal preferences when it comes to writing about, linkage and genetics and stuff. Largely because this is just the way that I was taught by one of my postdoc mentors. Let's just put genetic code. So if you want to write down an individual that is homozygous dominant, you could write like so, which is kind of the form that we've been using to date and probably the one that you're most familiar with. Now, if this gene is on the X chromosome, we could also write it like so. This being a female fly in this case. So now I'm telling you that not only do we have two dominant alleles, but that gene is located on the X chromosome. I mean, if we wanted it to be chromosome 2, we'd write it like that, right? So that's giving you a bit more information, but it's not. It's going to be on the X chromosome. So now we're telling you not just the genotype, but where that gene is located, because that's important. Now the other way, which is a little bit less easy to get, I guess. So we write it like so. This looks a little bit silly just like this because we only have one gene. And so essentially what we're doing is we're showing, well, this is one chromosome, this is another chromosome. We have a dominant allele here, we have the A dominant allele on the other chromosome. Kind of use these interchangeably, to be honest. They serve different purposes. And similarly, if you had heterozygote, make sure everyone can see that. You'd write like so, or like that. Another way of writing it is the fact that we don't need to worry about saying what the dominant allele is, because we don't actually care about the dominant allele too much. We just care about the recessive ones. So now you could actually write this as like so, where plus equals the dominant allele. You can also write it here. essentially saying the same thing, just doing it in slightly different ways. Now, this one matters a lot more when we're talking about linkage, because it makes for a very, very useful visual representation of potential linkage, right? And you'll see why in a second. So now if we had two genes, things get a little bit more complicated. We're just going to skip this bit. Now, if we want to show the relationship between those two genes, because that's what we care about in linkage. In a sense, you're getting a jump on the linkage classes that start on Thursday. So this isn't wasted time in any sense. So now it matters a great deal which chromosome these alleles are on, their relationship to each other. So now you can have, again, we're going to put plus as the dominant allele. It just actually makes the shorthand a little bit easier. Now we've got something real different. So what that's telling you, if you look at this, is that this chromosome, the one on the top, it's just kind of habit that the dominant is always put at the top. This chromosome has a dominant allele for each gene. This chromosome has a recessive allele for each gene. Essentially, this is a locus. That's the position on a chromosome which contains a gene which controls a trait. Back to some earlier terminology. This is said to be, the genes in this situation is said to be in cis linkage. And the reason why I'm spending a fair bit of time going through this is because otherwise you're not going to make any sense at all about what I'm going to do after this. So it's very good. It's like eating broccoli and stuff. Now you can also, it doesn't have to be like this. You don't want to make an assumption. You have to test this genetically. It could also be like so, where one chromosome, again, because we've got two chromosomes, diploid organisms, remember, one chromosome is dominant for A and recessive for B, and then you have the opposite. The other one is recessive for A and dominant for B. Both have the same genotype, but they're very different in terms of what those chromosomes look like. This is the language that I'm going to be using to talk about this stuff. Partly because it makes a lot more sense to me, so easier to write and easier to read, but also because it's going to be really, really important when we talk about linkage. Makes things a lot more visually accessible. Okay, anybody got any questions before I move on? Oh, forgot. This is called translinkage. These terms aren't super important right now for this lab. They will be terms that we'll be using in class. Okay, so hang on a sec, Erica. Sorry, Dipper. Let me just double check the deadlines. I will get to that, promise. I'm just doing the kind of fun stuff first. That is due next Thursday, the 1st of July. Because that would be seriously rude of me to ask you to write a lab report by this Thursday. I'd have a mutiny on my hands. All right. What was I going to say next? Okay, so, but please do interrupt at any point. So normally in a kind of a regular lab, one could call it such, I'd have a couple of opportunities to go over all of this a couple of times, but it's a little bit condensed. So this is the first lab of the linkage lab. Let's share this back again. So today we're just going over the experimental design essentially and some of the language. On this coming Thursday, oh, and also showing you where the data are. Move this out of the way so I can see what I'm looking at. This Thursday, we're going to go over how you analyze those data. In particular, how you'll generate a quality graph, right? So some of you have already been doing this, you know, generating good graphs in Excel. We're going to just kind of build on that and get you to do the analysis of the results, essentially, which is both the statistics and the graph making. We'll go through that this Thursday. And then I'll expect you to take the data that we have, which I'll show you in a minute or two, draw that graph, do that analysis, and that spreadsheet is due next Tuesday. So usually the due dates are the midnight, the lab following. So it's still sticking to that kind of format to give you time to A, do it, and B, ask questions in the following lab. And then in next Tuesday's lab, so a week from today, we will go through the chi-square analysis again, just because it doesn't hurt to do so. And we'll talk about in more depth the report writing and the, you know, the drive for that being an assessment in the lab. So everything we do has a purpose, but it doesn't hurt to actually tell you what that purpose is, because, you know, that makes you happier, generally speaking. But yeah, we'll go over all of those details in more depth as we go through this too. Does that help? Generally speaking, it's a little confusing to read the deadlines in here, but whenever you do a lab, the assignment from that lab is going to be due the midnight of the day of the following lab. So that's what this kind of means, you know, it's midnight of the day of the next lab period. Okay. All right, so question is, how do you determine whether or not two genes are close enough to be linked together? Right, and so essentially what we're saying by linkage is, are you more likely to find a particular combination of alleles together than apart? That's the kind of general theme that we're looking at here. And again, we'll go through linkage in quite considerable depth over the next couple of lecture days. You'll have math bleeding out of your ears. So this is going to superficially look like a dihybrid cross, but it isn't. It's called a back cross. Some of the principles are the same, some of them are not. And so really what we want to get is a situation where we have a dominant and a recessive version allele of each of the two genes that we care about in the same organism. So we want to end up with something like that. Because what we're looking for is recombination between those two genes. So if those two genes are genetic equivalent of miles apart, I don't know, let's say Oakland, California, and what's that place where Disney's at? Orlando, right? That's the equivalent of being so far apart, you're never going to be linked. Versus, say, San Antonio and Texas. Still a ways apart, but close enough that there is some linkage between them, right? So you're not always going to get recombination occurring between those two genes. And so that's the whole idea. How can we quantify how often recombination does and does not occur between two genes to figure out how far apart they are? And remember, and this is something that's going to be super important in the class as well. I'm even going to capitalize this and underline and an upside down exclamation mark, whatever those are called. And a few little stars. Recombination is random. And probability equals distance. So the further apart two genes are, go on, Robert, not too much longer. Should have had a stronger coffee from that Starbucks. You have some like weak ass tea or something. Oh, I know that was a cup of tea, wasn't it? Was that tea?
No, it's actually a hot chocolate.
Hot chocolate. All right, ask for like 2 or 3 shots of espresso in that next time. It'll both be delicious and keep you awake.
Yeah, noted.
Yeah, it's tough. It's tough staying so focused for these intensive days of learning, and I try and do my best to kind of take that into account, but we still do have to cover a whole bunch of stuff. So this is the most important principle you're going to learn for linkage. Recombination, this is random. So if you randomly, I don't know, fire a BB gun, right, at a can, your likelihood of going between two cans is going to go up the further apart those cans are, right? If you just shut your eyes and fire, obviously you shouldn't do that because that's bad etiquette, but if you just shut your eyes and fire off randomly, the probability of going between two cans is going to be equal to the distance between them. That's what linkage is all about. It's all about probability equals distance. Smaller the distance, the lower the probability recombination will occur. Okay, cool. Doesn't hurt to kind of bang this into you over and over and over again, because once you get this, it's easy, the rest of it. Okay, so we need to end up with an individual, female, because this is X-linked, remember, you only get recombination between two X chromosomes, not between an X and a Y. We need to get a female who's heterozygous for both of these genes, because that's where we're going to see recombination occur. So step one is to create said individual. We're going to do that with a homozygous dominant female. And you know it's female because this is an X chromosome and you got two lines. Right? So kind of in other words, your genotype is that. If you can see that, kind of tiny, sorry. I'm a lot closer to this than you are. So this is going to be a red-eyed brown body. This cross is all laid out in the linkage, shoot, what's it called again? You know, sometimes I can't even remember my own name. What is it again? There we go. So in the lab 5 to 7 linkage project folder and course content, the first file, the linkage fly project, contains all of this stuff, right? So I'm going to put these pictures up after class finishes, and all of this is already in there anyway. Just going through it with you. And some of you will probably wonder how your lab reports can be structured. I'll talk about that with you in a couple of labs. But there's also a long form and a short form rubric in the Fly linkage, whatchamacallit, too. Just here. If you're wondering where these came from, I only put them up before class because I realized that they weren't there. So these should contain everything you need to know. If not, we'll cover it in class. And if that doesn't work, you can just ask. Okay. Another fun thing, I'm not sure if this will make you feel better or if it'll make you feel worse. I've went through something not quite the same as this, but similar kind of stuff with a PhD student recently who was doing her first genetic cross. So these are the kind of skills and ways of thinking that will stand you in good stead if you follow this kind of stuff into a career as well. Okay, so this is our female, and we're going to cross, which is actually an X rather than cross. That with a hemizygous recessive male. And we know this is a male because it has a honking great big Y here. That's its Y chromosome, right? There's no alleles for A and B because it doesn't have those genes on the Y chromosome. It just has a Y chromosome. It's like, meh, nothing much. On its X chromosome, it has recessive alleles for A and B. All right, just make sure you're not all asleep, and I wouldn't blame you if you were. But what possible gametes are we going to get from these two individuals? Remember, I always bang on about if you want to do a cross or a Punnett square or whatever, you got to figure out what your gametes are going to be. So what gametes are you going to get from these two individuals? What possible gametes? So I'll give you a clue. The male, one of your gametes is going to have a Y chromosome, right? Yeah, the cross is, so this is the genotype of one individual, Galen. This is the genotype of the other individual. The big X between them means that we're going to be mating these two individuals together. Again, that's kind of slightly annoying genetic shorthand. You just get used to it. Good question, though. Okay, so if we're going to be segregating these chromosomes randomly into gametes during meiosis, you're going to get a gamete with a Y chromosome in. What's the other gamete going to be for this male? It's going to have an X chromosome. What alleles is it going to have on that X chromosome? Good answer there, Galen. What alleles is that X chromosome going to have for the A and B genes? Sometimes it helps if I, that's even worse. Kind of draw it like so. So this one line is the X chromosome, the other line is the Y chromosome. Desiree, 100% correct for this individual. So this homozygous dominant female can only produce, because it's homozygous dominant for both of them, right? Gametes with two wild type alleles on the X chromosome. So yes, good answer. What other gamete can you get from the males? One chromosome is the Y chromosome. The other chromosome is the X chromosome. But what are we going to have for alleles on the X chromosome? It's written just there. Almost. It's going to be or another way of writing it. Try and do this in different ways because everybody kind of interacts with this information in a different way. Does that make a little bit more sense? Cool. I know it's kind of, ooh, you could almost classify this as some kind of torture to be talking about genetic crosses after already having 2.5 hours of genetics class. But once you get it, it'll be a little bit more straightforward. Okay, so while I take a picture, have a think about what the offspring of those two individuals will be. I'm just going to write another line there. Doesn't matter whether there's one line or two lines. Sometimes it just helps make it a little bit more visually accessible. Okay, so we could do this as a Punnett square, which is like the surefire way of figuring out what your offspring are going to be like, right? So from the mother, we have plus plus gamete. From the father, we have, oh, I hear a snuffling puppy, a X chromosome and a Y chromosome. X chromosome has our recessive alleles. Where are you at? Oh, thank you, pup. Just dropped a load on one of the puppy pads. Nice. Clean that up when I'm finished. What do you want? Hang on a second. Puppy lifting. You want to come up here? That squeaker doesn't work on that anymore. So put this together. You will get our wild type chromosome from the female, our mutant chromosome from the male, or you will get our wild type chromosome from the female and a Y chromosome from the male. So you're going to have heterozygous female or dominant male. Does that make sense? Because you can only have one possible gamete from a homozygous dominant individual. That's going to be a dominant allele or two dominant alleles if you have two genes. And from the male, it's a hemizygous recessive. So it has recessive allele for each gene and a Y chromosome. So it too, well, it can produce two possible gametes, one with a Y chromosome and the other one with an X chromosome with recessive allele for both A and B. Any questions? It'd be worthwhile, because again, this is a lot of stuff to take in, particularly when you're tired. Maybe tomorrow, maybe on Thursday, just go through this stuff in the fly linkage report file and see if it makes sense. No, you're not having one of these, sorry. And so remember at the beginning, what we need are individuals that are heterozygous for both genes. That's these ones. So we're not going to cross these two. We're not going to cross the siblings with each other. We're going to keep these and we're going to throw away these ones because these are the individuals in which recombination may occur. Right? Because to be able to see recombination, you need to have individuals that are heterozygous for both of those genes. I'm actually going to take a picture of the puppy first. I have like a puppy picture of the day thing going on. Okay, right. So now things get a little bit more complicated. Sorry. So now we have our second step. We have our heterozygous females. These are the females we got from the previous cross, the dominant parent female and the recessive male. Now, what we want to see is recombination occurring between these two? And so if no recombination occurs, what you'll get, oh, that's kind of a sad look. Unintentionally, I have to say. Not sure that one looks any better. It's like it's in something bad. If there's no recombination occurring at all, the only chromosomes you're going to see in the gametes are the parental chromosomes. That's another important term. If recombination does occur, however, what you'll get, and I'm going to use a color here, is this chromosome crossing over here to join with that chromosome. So you're going to get plus, and then you literally follow the line B. That is a chromosome that you don't see in either of the parents that generated this individual. Right? Because recombination has occurred between this chromosome and this chromosome during meiosis. This is called a recombinant. And if you have this one, oh, it's a hard life, dog. You have to have the opposite, because if it goes one way, it's got to go the other way. So if you have plus B, you have to have A plus. These are our recombinants. And if recombination occurs every time, what you should get is a 1-to-1, hole in the board, to 1-to-1 ratio. If recombination occurs every time. Now, question, and it's going to be a little bit of a oh, my brain hurts, professor, kind of question. What genotype are we going to need to cross with this individual? It's got to be a male, right? Because, you know, females can't reproduce themselves. What genotype male is going to have to be crossed with this so that you see these different combinations? Let's put it a different way. If I used a wild type male, red eyes, brown body, this offspring would be homozygous dominant for both, right? What phenotype would this one have? It'd be plus B over plus plus. It'll be dominant, dominant. What phenotype would this one have? It'll be A plus over plus plus, dominant, dominant. What phenotype would this individual have? It'll be AB over plus plus, dominant, dominant. So if our back cross fly was wild type, you wouldn't see any difference between all these possible gametes. Think about test cross. What genotype does your tester have to have? It's kind of going back to exam 1 stuff. What genotype?
Would the male have to be like a homozygous recessive to?
It would have to be recessive. It wouldn't be homozygous because it's a male, so technically it would be hemizygous.
Yeah. Right, but it has to be recessive. Yeah, recessive. Yeah.
Exactly, and thank you, Galen. You got that too. It would have to be recessive. So it'd have to be that genotype. So when you put them together, you'll be plus plus over AB. That would be dominant, dominant. Plus B over AB would be dominant B. A plus over AB would be A plus, and then you'd have AB. So you'd be able to see the 4 different possible combinations you can get. Is that classroom automatically turning the lights off on you?
Yeah, like every 15 minutes it just turns off and we have to get up and.
Super annoying. One way to get away from that is to, how did I used to do? I used to have this problem in cab. I think I used to have to close the like the blinds.
They're closed.
Oh, well, maybe that didn't work. I don't know. Gets you some exercise, I guess. Blood flow to the legs and stuff.
Yeah.
And so what you would be doing if you're going to be doing with this with your own flies, I'm going to give you the data, but what you'd be doing is then you're going to be counting the relative proportion of these 3 different groups. Dominant, dominant, dominant B, B dominant, AB, right? Or recessive, recessive. And you'll be seeing how many recombinants do I get? Because if my 2 genes are linked, they're close together, I'm going to get fewer of these recombinants than I'm going to get of these parentals. That's the basics or basis of this whole experiment. Now, I'm not going to ask you for questions because probably right now, like your brain's like, oh, and your eyes point in different directions. But this should kind of give you an overview of what we're going to be doing. Right. In that document, let me show you wherever it's gone. Here we go. Bring up the trap. So essentially, this is exactly what I've just already told you. I just used the letters for the actual genes, right? So the cross plan is all there for you. You can go over it another time when you've got a clear head. And the data that you would have generated, actually, these are fabulous. These are really great data because there's a huge amount of individuals that have been scored. Sorry, I'm not immune to tiredness. And these are the white-eyed, yellow-bodied, red-eyed, wild-type bodies, and so on. Okay. So these two are your parental combinations. It's what you see in the parents, hence parental. These two combinations are at far, far, far lower counts. So immediately that tells you that these two genes are most likely linked because you have an excess of parentals, which means that recombination is not always occurring every meiosis between these two genes. And so we're not going to go over it today. We'll do it next lab, Thursday. Yeah, we're going to go over this on Thursday. How to analyze those data, right? How to, oh, got the wrong file. How to put them together into a good graph and do the other analysis. There are other, we also have a really cool little video kind of walking you through doing the graph, right? So the graph is worth 100 points. So that's what, 1/8 of your lab grade. And it's really not too hard, particularly if you just watch this and follow the instructions. But most of you, I'm sure, will be able to do it anyway, even without that help. And then next lab, we'll go through some of the data analyses, the chi-squared. Again, it's still laid out here for you. I don't think I've used the same data. I'm not going to be that helpful. But you have the work through. There's another test, the odds ratio and map distance. Again, don't worry too much about those right now, because we'll be going over that in more depth next lab section. And then your other assignment from these 3 labs, again, today is really just like an intro kind of overview type deal, is a 2 to 3 page mini lab report. You'll be using the same graph that you supplied for your first assignment, and you'll be including all these other analyses. Bit of an introduction, bit of a methods, bit of a discussion, all kind of laid out here. There's your mini lab report. That's worth 200 points. So that's a quarter of your lab grade. So this is a relatively straightforward way of getting a very large chunk of your lab grade, which is again a decent sized chunk of your course grade. And one of the best ways that you can lift up your overall grade for genetics is by doing very well in the lab. It's kind of the best way, other than, you know, studying your ass off, obviously. That also helps. Now, because this is a compressed, like twice the speed semester, there's not a whole bunch of time to get feedback. You know, actually, you know what? I'm going to change this. I'll upload this later. I'm just thinking about it. These things are not kind of cast in stone. Now, downside is that's going to be over the July 4th weekend, but so I do like to be able to give students the opportunity to get feedback on their lab reports. It's a really useful part of the process, and it's no different to what I do with people that I write papers with and so on, right? Your draft is always going to be better the second time or third time round rather than the first time. So this mini lab report, if you can get that to me in draft form by Thursday, Friday of next week, I promise you I'll give you feedback on how to improve it with enough time for you to be able to submit it by the 6th of July. And usually when people do that, as long as they do the things that I recommend they do, get very good grades for their lab report. It's not hard to do so. You just need to make sure you do all of the things that I'm asking you to do, which are kind of laid out here. Again, we will go through this again in a third lab, you know, just to give you a chance to kind of percolate over it and think about stuff that you want to ask about. That's pretty much it from me. So all of the information, I mean, you didn't even really need me to sit here and tell you all this stuff, but all the information that you need is in these 3 documents. My aim is to help explain some of it and, you know, give some tips and tricks and stuff. Cool. Does anybody have any questions? Is anybody capable of asking any questions? It's probably the more.
So what do we have due for next lab?
Nothing.
Okay, because this was just all info and stuff.
Right.
Okay.
I'd recommend reading through that linkage fly project file again. Most of it will tell you what you already know now after going through this, but it will help reinforce that. You're obviously sitting in the wrong place there, Robert. And bring any questions you have with you, if you have any, to lab on Thursday. Yeah, but nothing specific from today. Cool. As always, if you have questions, email me them or save them for next lab and we'll go through them then. And all of this stuff will be up and posted after I've had a cup of coffee, which will probably be starting in about an hour. Takes me a little bit while to kind of become functional again after I finish teaching. Cool. Well, thanks very much, everybody. It was oh, hang on, question. See you, Marcus. See anybody that wants to leave? I just got a question about the corn chi-squared question lab. The question say describe the information we got. Do we do it verbally or show problem solving? Give me just a wee second and I'll try and pull up the question so I know that I'm answering the right question. Oh, I think I know which questions you're referring to. So, for example, in, I think it's question A4, I ask which phenotype is dominant, right? And then how do you know that it's dominant? And so I want to see, how do you know that that particular color of corn kernels is dominant, right? What do you need to look at? You could either do it from like first principles. Well, I know it's dominant because it has this genotype and this genotype equals dominant. Or you can go and say, well, I looked at the parents and the F1s have the same color as this particular parent, therefore this particular color is the dominant color. It's essentially the two different ways you can do that. But I need you to write that down for me, and so I can see like the the logic process that went on to generate that answer. Cool. All right. Anybody have any other questions? Okay. Again, if you don't think of them by the time I click the end button, just email me them, particularly for the assignment today. I'll be on my computer until probably dinner time. And for the other stuff, we'll talk about it next lab period. Cool. All right. Thanks very much, everybody. Enjoy the rest of your day, and I'll see you on Thursday.